There we go. Sorry, a little delay on my microphone getting picked up, but hello, good morning, afternoon, evening, and welcome to Friday. Here, there we go. I had to take some of that stuff off because it was wrong and I left it up from the last stream. But I hope you're doing well today, and thank you for joining me at the bench. Here, I'm gonna actually switch over to our regular playlist while I'm at it, which I didn't realize that it was set on shuffle, so you guys might have heard some different music for my regular starting one this morning. It's all good, right? It's all good. But we'll get started right off. So, good morning. Yes, so today we're continuing with our enrichment project, so our targeted DNA project that has been going on for a while now. I'm going to be doing 5 enrichments today. 5 enrichments. So that's why you can see there's a couple of different tube layouts than before. So we're going to do that today, and I am not completely ready to go like I usually am, so starting off today I actually need to check the concentration of one of the genomic DNA samples that I'm working with today. So we're going to start off that instead of just going straight to the master mix that I usually do, so it's all good. It's a Friday. So yeah, thanks for joining me at the bench. I hope we have a good day for a stream today. The internet stays stable. That would be great. And yeah, let's get right to it. So I'm gonna put on my lab coat as always, safety first. And we will get ready to check the genomic DNA sample that I have. Alright. Lab coat on. Here we go. Now I'm going to put on my gloves. I actually am going to make a dilution of our genomic DNA because our one kit that we use and has been back ordered forever still hasn't come in. It's coming in today though, so we won't have to keep doing these dilutions. I'm excited, but it's basically, so usually I do genomic DNA checks before stream just so I'm ready to go. You guys don't have to wait for that. I'm also going to grab this because we're going to make master mix. But I'm just a little behind this morning, but that's okay. So you can see a little more of behind the scenes for the streams. So this is a new sample that was prepped yesterday. First things first, we need to get everything set. I'm going to go grab the tube that we need and the dye. Let's see. Tube, and here's the dye. But at least I was on top of it and got the buffer out to thaw, which is good. So here's the dye, here's our tube. And I'm going to add 199 microliters of buffer that we need. So that's it. There we go. And we need 1 microliter of the dye. And then I will make our 1 in 10 dilution of our gDNA. Okay, so 9 microliters of water. There we go. And just stick it in this tiny little tube right here. Whoop. See, I'm already dropping things. And microliter of our channel. Oh my, which is very. This is. I got a feeling it might be high. I hope I don't have to do another 1 in 10. I hope I don't have to do another 1 in 10. I'm gonna mix this up. Spin it down briefly. And put a microliter of this into the tube. And then we let this sit for 2 minutes after I do a brief vortex, let the dye incorporate into the DNA, and then I will measure it on our cubit so that I know how much we have. Alright, 2 minutes. 2 minutes, let's see there. Nope. There we go, there we go. I'm gonna go put this back. We don't need this right now. And then while we're waiting, I can get the other samples that I have ready to go. Because I already have concentrations on those. So that'll be nice. But just a second. Alright, let's see. What do I need for the other 2 samples? I have to go back to my lab notebook where I figured this out. I need 53 of one. And just double check and make sure that I grab the correct one. This one. That one. Alright, and then the other one is 30. Alright. 30. That one's all set, so now we just need to wait for a couple more seconds. And then this one will be ready. Then I'll go measure it, and I'll do a little bit of math off to the side so I know how much I have in there. Alright. Okay, let me go do that. I'll be right back. Alright, so it was still too high to check, so I need to make another tube. So let's bring this back up. So we'll do a 1 in 20 dilution. And hopefully, it'll be okay. And 1 in 20. Buffer. So I'll have to wait another 2 minutes. Another microliter of dye. There we go. Just mix this up a little bit. Okay, so let's add water. Alright, so this is going to be 1 in 20. And we'll take another microliter of this. And check it in 2 minutes. Hopefully, we will get a good measurement on this. And I have an idea of how much we actually have. Alright, let me mix this up. Another 2 minutes. Alright. Okay. While that's going, I'm going to put this back again. Since the other ones are going, at least I know how much water I can add to our master mix for the other two, so I will do that. So it's going to be... We need 80, I'll do 86. 86 water. I'm just going to add it to our master mix tube right here. So that's set and ready to go. And then let's see, I'm going to need 50 microliters of our buffer that we're using for this actual mix. So remember, the very first step in this process is to dephosphorylate our genomic DNA. And that is because anytime that the DNA is fragmented, it leaves a free phosphate at that broken spot. And we don't want that when we are starting out, because downstream we're going to be adding our Cas9 protein, which will cut at the specific spot we are targeting in the DNA. And we want those phosphates to be free so that we can actually sequence those. So we want to get rid of anything that is broken right now, get rid of that phosphate so that that doesn't basically get sequenced when we're doing downstream. We just want to hopefully sequence just what the Cas9 cuts for us. So that's what this initial step is for. And then, oh, perfect. All right, so second time's a charm. Let's see. Hopefully, this will give me a concentration. I'll be right back. All right, yes, I got a concentration. All right, let me do some math here. Oh my gosh, that's a lot. We gotta get a pretty high concentration there. So a little more math off to the side. I need to figure out how much I'm going to be using in this reaction. Just a second. Alright, yes, so plenty. We have plenty of DNA to work with. Alright, so actually need this one. And I can show you how we make our tips. There we go. So just use, because we don't have wide bore tips, but we use these tips instead. Just cut it. It's perfect. Alright. So I'm going to add this genomic DNA. That's pretty viscous. We do not need a lot of this. It is pretty high concentration for what we're doing today. Which is fine. I'm glad about that. I'm happy about that. That means we'll have a lot left over. Okay, so that is done. Alright, so now I need to add the water for this one. Oh gosh, it's a lot. Ooh. Not sure if my tube's gonna be enough. I might have to stick it in my centrifuge tube instead. Just because this one requires a lot of water. So, you know what we're gonna do? Let's move that over to a bigger tube. One of the fun things about science molecular biology of the bunch. Let's move these colorless liquids into another spot. There we go. That one's all set. Bring this back to 30, because that's how much I'm going to need to add of the enzyme that's going to remove those phosphates for us. There we go. And then for this one, I'm actually going to need 240 microliters of water. I'm going to take it from my big water. There we go. Alright. That's all set. So we have our water, we've got the buffer added, we've got our genomic DNA. I can get rid of that. So now I'm going to go grab the enzyme from the freezer. I'll be right back. Alright, so we have our enzyme in our little caddy right here, called QuickSip. We need 30 microliters. There we go. That's right to the master mix. Now, I just need to mix this up. And we will aliquot it into our tubes. So, let's see. This one's going to be 90.8. Okay. Trying to save on plastic, but I reuse the tip. Go. And then the other ones are going to be 70. Next one, master mix, and then whatever's left goes into the last one. This one, whatever's left will go into. Perfect. All right. Which whatever's left was it would have been oh, math. Oops. 46, yeah, 46 microliters. Okay, so now I'm gonna mix these by doing this. So we do the inversion so we don't fragment it anymore by pipetting or vortexing. That's a little too rough. We want to have long DNA strands as we can. And then I'm going to put this into our PCR machine. It's going to incubate at 37 degrees Celsius for 30 minutes to let the QuickSip do its thing, dephosphorylate any ends there, and then 80 degrees Celsius for 2 minutes to basically kill the enzyme, make it stop doing what it's doing. It'll denature it and won't keep going forward with it. So I'm putting that into our PCR machine right now. And then while we're waiting for that, we will get our Cas9 all set. All right, it's running now. So let me put our enzyme back. And actually, let's set this. 32 minutes. All right, 32 is working. That's the only problem. It doesn't. Oh. Yeah, it's not gonna stay. That's okay. Oop. Put it back. There we go. There we go. Set that 32 minutes. All right, let me grab what I need to, and I'll be right back. Alright, I am back. So now we're going to get our Cas9 ready to go. So this is going to be the protein that's going to cut our DNA in specific spots. So this is what's going to be targeting all of our stuff. Let's see. So what we're going to do is we're going to add the protein, and we're going to add the oligo that has the specific target in the genome that we're looking for, and basically put them in the tubes together, and it's going to incubate at room temperature while our genomic DNA is dephosphorylating, and it will basically complex together. So the first thing I'm going to do is add the water that we need to our tubes, and each of them is different. So let's first start with the 3 different. So this one right here is for 3 different ones. So we're going to be cutting in 3 different spots, and this one is for 2 different ones. Let's see, I need 12.2. Here's the first one. Water. Goes there. Next one is 11.7. And then I want to say, yeah, 11.4. That one is for this one. Then next one is 10.9. This last one. And the other one needs a different one, needs this one. And that is 8.5. Now the water all set and ready. Now the next one is we want to add the buffer. It's going to be 1.5 microliters of buffer for each of the tubes. It's this one right here, same buffer that we were using before. I'm just going to reuse the tip because it's just water that's in here. I've not added the protein or the oligos, so we don't have to worry about contaminating our tubes. Alright, so the buffer's all done. That is all set. I'm gonna leave that there. So the next thing I need to make a dilution of our Cas9. Now I have to think of how much Cas9 I'm gonna be using. So 1, 2, 3, and then for this one, 2, 4, so 7. I need 7 microliters. All right, I'm gonna do that. Math, I love math. 1.4. Alright, okay, so I need 5.6 of our buffer, so we make a 1 in 5 dilution of the actual protein that we use. 5.6, I won't worry about it. Yay, math. And this is the buffer that we use right here. Right in this one, and then I need 1.4 microliters of our actual protein right here. There we go. Mix it up, spin it down. Alright, so now I'm going to add the protein. I need 1 microliter in each of this one. Okay. And then the other tubes are going to get 2 microliters. Microliters. Perfect. Alright. Well, that's done. So I've got a hair on the flip. Alright, so that's done, and I'm going to put this back, and this buffer back, and this buffer back. Now I'm going to grab the actual RNA, the sgRNA that we're using. Alright, I grabbed the sgRNAs that we needed, so we'll be all set for that. Oh, I just realized. What did I do? I know what I did. I keep thinking that we only have one sample. I need more buffer. I need more buffer and water in the other two. Ugh, it's a Friday. It's a Friday! I need enough to make it for 2 samples, not just one on those. I put the right amount of Cas9 protein, but my brain said no on the other things. That's alright. Easy fix. Easy fix. We just add more. I think I did this last time too. I'm telling you. At least I fixed it. Oh, welcome to the lab. Alright, so this one needs 11.4 water. And then the other one was the smaller one. And that is, I just want to make sure I have the right one. This one. Yeah, this one's 8.5. 8.5. There we go. Alright. There we go. Okay. Solved. Okay. Time to add our guides. So basically the guides are for the spots that we want to cut in the genomic DNA. And 1.6, let's do that one. 1.6 on this one right here. Make sure that's thawed. Yes, it's thawed. Alright. That one's ready. Then let me double check on this one. So the next one is 1.1, or actually, no, that's for 2. So that'll be different. Let's keep it to this one. 0.8. 0.8 for the next one. That one's done. And I'm actually going to grab a different pipette for the other one, because this one is going to be pretty low. And this one is much better at going lower. This 0.3. So, this is much better pipette for going that low. There we go. So those are done. Now, remember, this is going to be for 2. This is going to be for 2 right here, so 2.2, or actually, since we're up so high, let's do the highest one first. There we go. And it's going to be this one. Which I think, yeah, basically finishes off that one. To make more of that if we need it. And then 2.2 on this one. Ugh. Alright, there we go. Those are all set. I'm just gonna mix these up. Get them going. Spin them down. There, perfect. Alright, so these are just gonna sit here and wait for the next 20 minutes, and I can see that we're already having issues with the internet, unfortunately. We're dropping frames. But that was just at the beginning, so it looks like it was only like 20 minutes ago. So looks like it's back to being stable again. We had a little bit of an issue. Hopefully we're back. I'll have to check and see. But until then, I'm just going to put this stuff together, get what we need out next, put back what I need to, and we'll be back in 20 minutes. Alright, so we're almost done with this incubation for the dephosphorylation of the DNA and our complexing of the Cas9. So if you saw me doing some stuff in chat, I was checking to see what our experimental stats, if I had updated them or not. So I have since updated them with 2 more regions. We're actually correct with the timer, but the experimental stats was 2 off. So yes, we have done 64 of the 73 that we currently have. And this will be 65 right here. So I'm just setting up the timer for our next jaunt, which is going to be a 40-minute incubation after this. And I'm just waiting to hear from our PCR machine. Will beep once it's ready. But let me also, we don't need to see this right now, don't need to see this right now. There we go. But I hope you are enjoying the chill music and everything so far. Let's see, PCR machine, you can do it. 30 seconds, 30 seconds. Okay. So yeah, once the PCR machine is done, the next step is we're going to actually do the cutting with Cas9, and that's what we made this for. So the Cas9 is going to target the specific regions that we have set up in this complex, and then once it cuts, we're going to add a enzyme called polymerase, which will add an A, so the nucleotide A, to those cut sites, and we need that A in order to have the sequencing adapter ligated or added to the end of that in order to do the actual sequencing. Alright, so these are ready. Here are our samples. I'm going to move these up here. Move that off to the side. I'm going to go grab our little enzyme caddy again so that we're all set with the polymerase. Alright. Little enzyme caddy. All set and ready. I'm going to let these cool down a little bit because they were just at 80 degrees Celsius. little too hot to add the Cas9 and the polymerase to right now. We don't need to denature those or kill them right off the bat just because our samples were too warm. I just let these hang out like this. But yeah, so that's the next step. Let me get this ready. This needs to be set at 1. And this one needs to be set at 15. There we go. Oop. I almost dropped it. Let's not do that. Free up a little more space here. Move you out of the way. Don't need you right now. There. There. And then once the next incubation is set, I'm going to be grabbing some buffers that we need for the actual library prep, since that's what we're going to be moving to next after Cas9's done cutting. And I will also get some tubes set up, because we're going to be moving out of these PCR tubes into microcentrifuge tubes, so I'll get the label. But this will be our longest incubation coming up. Let's see. Ah, that one's ready. And that one's good. How about you? Yeah, you feel good. Alright. So I'm going to add our Cas9s first. Add this one. Right, those ones are set. Now we're going to add this one to each of these. Alright, that's Cas9 added. Perfect. Now we're going to add our A nucleotide, so DATP. 1 microliter to each of these. Alright, that's done. Now I need to set this to 1.5 microliters. And we're going to add 1.5 microliters of our polymerase, which is hanging out right here. And this is the one that's going to add those A's to the ends of any cut DNA from our Cas9 so that the sequencing adapter can ligate in the next step. Alright, so that one's almost... Ooh. Alright, nope. Too many touches. Too many touches. Let's not outside contaminate anything. Alright, there we go. Last one. Alright. So that is set. I'm going to mix these up again. And then these are going to incubate for half an hour at 37 degrees Celsius to allow the Cas9 to do its thing. And then 10 minutes at 75 degrees Celsius to let polymerase add those A's to all the cuts that Cas9 should have made during this time. And that's all done in our PCR machine. Spin it down. And I'm going to pop this in our PCR machine, get it ready. Then we will hang out for 40 minutes while I get buffers that we need for our next jaunt, which is in the library, sequencing library setup, and making tubes, labeling tubes. Alright, so that's all set and ready to go. So unless you have any questions, we will just hang out and enjoy the music as always. Alright, so we only have a little bit left on this incubation, so I came back a little early. I got all the tubes that we need set for our next step that we're going to be doing, which is getting the library adapter ligated. And I got our buffers for the next step also thawed, which is good. Move some of this stuff over since we're going to be using this next. And I also made a new buffer that we have right here for the next step because I didn't have enough. So I just need to transfer it over. And if you have been on here before, this is the buffer that is very viscous because it has polyethylene glycol in it. So you can watch how slowly it's stuck to the. The only problem is sometimes it gets stuck to the tube. Because I added a cold solution to it, so now it's gone on the solution just a little bit. But that's alright, it's not going to be a problem. But as you can see, very viscous. It comes up through the tip. It takes a long time. There we go. There we go. I just need to move this over. It's gonna turn off. Got that. Alright, so that's done. Waiting for the PCR machine to beep. And I'll just grab the rest of this buffer. You're good. That's fine. A little at a time, it's alright. Oh, there goes the PCR machine. Alright, alright. Pretty mixed up the plant. See if I can get the rest out of it. There we go. Okay. Let me grab the samples out of our PCR machine. Okay, so sets. Bring them over. I'm gonna go grab our enzyme caddy because we have the adapter and our next protein enzyme that we're going to be adding, ligase, in here as well. Alright, so here we go. Okay. So, first things first, we need to move all of these into the microcentrifuge tubes. So we're moving to bigger tubes this time. I'm just going to bring it to a arbitrary volume with our homemade wide bore tips. There we go. And then I need to change up our what's happening now graphic. We don't need that right now. Do that before I forget. There. Alright, move the other ones over. There we go. Last one. Perfect. Alright, that over there. Alright, so now I'm going to add 25 microliters of water. So once all my components are added, then it should equal to 200 microliters. Make sure, yes, sorry. I wanted to make sure that I had what I needed in there. So each of these is going to get 25 microliters of water. Then we're going to add 50 microliters of the viscous buffer, which we call LNB, to each of these. And what this buffer does is, like you saw how viscous it was when I was pipetting it into the tube, is it basically is there to take up space. So it'll get all of the components in the tube closer to each other, so we have a much more efficient reaction. And that reaction is to put add the adapter to our genomic DNA that we just cut with Cas9 at specific spots in the genome. And then we had the polymerase add an A nucleotide to the end of those cut spots. And at each of those spots, we're going to hopefully ligate the sequencing adapter, because the sequencing adapter has a T overhanging the end of it, and A pairs with T. And so this polyethylene glycol solution will hopefully get everything closer together, so the adapter is more likely to come in contact with our detailed genomic DNA. Well, target now to hopefully Cas9 cut targeted regions in the genomic DNA. Alright. I'm just being slow with it because it is so viscous. Pipette slowly. Leave it in there for just a little bit so that make sure it's all in the tip. There we go. Alright, and that is done. So now we're going to add 5 microliters of our adapter, or library adapter, to each of these. I'm gonna have to get more tips. I'm gonna be one off. Oh, one off. So each of these is gonna get 5 microliters of our sequence adapter. I'm going to have some more of our flipper chips. Alright, let's grab more tips. So this one is all done. Empty. There we go. Alright. There we go. New set of tips. This will be our last one. Alright, now we're going to add 5 microliters of the ligase, and this is the enzyme that's going to add the library adapter to our cut DNA. And it's 5 microliters for each of them. Alright. Looks like it's basically the last one of that tube. Spin it down. See if we can get more out of it before I move on to our other tube that's in there. Yeah. Nope, there's nothing in there. That's fine, that's fine. Got another one right here. Just wait. Alright. Perfect. So that is set. Okay, so what we're going to do is we're going to have this incubate at room temperature on our loop rotator for half an hour. Give it plenty of time to do the adapter addition to our genomic DNA. We have another semi-long incubation, half an hour. So let me get our tube rotator. Just a second. Alright. So I got a tube rotator. Let me set this to 30 minutes. And then actually I'm going to get the rest of our timers set up because we need 10 minutes. Series of 10 minutes. Okay. Add the tube. Put it too close. Knock it off. Alright, there we go. 30 minutes. Let that go. I'm going to put away some stuff and you guys can chill out with music. Alright, we're coming up on the end of our library adapter ligation incubation here. So we'll get ready for the next step. Put my gloves back on. Alright, so next we're going to add some TE buffer, and what that is going to do is it's going to stop the ligase, and if the Cas9 is still going, stop that Cas9 from doing anything else, and get basically the DNA ready for cleanup. And get that out of the way. You guys can go here. And when we do the cleanup, we use magnetic beads that will attach to the DNA and help us pull it away from the buffers and the proteins that are already in here and get it in the correct buffer for sequencing. So let it finish up right here, and this is going to be a series of, we'll do 10 minutes on here after we add the beads and the TE. So it'll be another 10 minutes, and then after that we're going to clean it up on this magnetic rack. So that'll be all set. And it'll just be a series of that, and we'll be using this buffer right here. This is our cleaning buffer, long private buffer. Clean it up on the rack. It'll be all set. Almost done, we're getting there. Just a couple more seconds. Alright. I'm gonna set this at 10 minutes. Wait for it to come back around. Turn this off. Alright. Okay. I'm stuck in there. Let's move this out of the way. So I have a little more room. Let's add the TE to all of these. I have to put more in here. Almost out. Alright. We'll mix it up. Then I will add magnetic beads. And we'll let it rotate for 10 minutes. Let the beads collect all the DNA and get those ready for cleanup. Right, mix it up. Looking good. These look good too. Nice, looks good, looks good. Alright, let's add the beads. So it'll be 120 microliters of beads. There we go. Let's vortex these. Just make sure that the beads are thoroughly resuspended. And you see they're brown. There we go. Alright, so each of these gets 120 microliters. Alright. I know I'm gonna have to open up the next one, so might as well get that one ready. Because I don't have enough for set. There we go. Just add that. Perfect. Perfect, perfect. Get rid of that. Okay. Got all the beads added. Do a little bit of a mix so you can see that now we have a nice little light brown color to our samples. I'm going to put it back on the rotator. And then 10 minutes rotating. And then we'll start the cleanup. All right, I'm back. Okay, so this is almost done. And then we're gonna start our cleanup. All right, move that off to the side. I don't want to wait until this comes back. There we go. All right, so I'm gonna put these on here first. Get this set done first. And now I'm gonna move you guys over here. Get this out of the way. We don't need this anymore. It doesn't take up space anymore. There we go. Off to the side. Ooh, I might be able to grab another rack. Let's see if I can grab another rack. Yes, I was able to grab another rack. So, what we're doing here is we're cleaning up this reaction. We added those beads. The beads should have bound the DNA, and they're magnetic, so I'm putting it on this magnetic rack. What you'll see is the light brown solution is going to become clear with basically the beads showing up down here. So that's all we're waiting for right now, is for all the beads to get close to the magnet that should have the DNA. And then we'll start cleaning up these with, I use 150 microliters of our long fragment buffer here. First, we're going to start off with the big one, just to make it faster in that case. So it'll be easier for all the downstream cleanup. So we wash twice with a long fragment buffer. And I have the big one just because we're going to be doing, we're going to go through probably, you know, a good amount of that. Alright. It's getting there. It's still a little cloudy. I'm waiting for it to get, like, mostly clear. Because it's kind of hard probably for you guys to see, but it's still a little fuzzy in that not all the beads have gotten to the magnet yet. So I want it to be pretty clear before I do it, because I don't want to lose any of our DNA that's in the solution still, because the beads aren't there. So a couple more minutes, or maybe a minute. This one is almost there. These two need a little bit more time. Same with these two. So let's give it a little bit more time. Move this over here. Put that there. 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 Much better. Much better. Oh yeah, that looks a lot better. That looks a lot better. Alright, these are ready to go. These are ready to get washed. Oh, there we go. So what I'm going to do is I'm going to carefully remove the liquid without touching the beads, because we just want to remove the liquid. We don't want to lose any of our precious DNA that we have on here. I'm just basically trashing it. We don't need any of this stuff. This is all the protein that we had left, and the buffers that we used in the past, which would be very detrimental to our downstream sequencing. So, there we go. One more. Oop. Perfect. Alright, that's done. I'm gonna move this off to the side. I'll do that for later. Alright, so I'm going to add 150 microliters of our long fragment buffer to each of these tubes. This. Alright, close it up. Close these up. And then mix it up just a little bit. Just a little bit. And I spin it down. And then just do same thing. Put it on the magnet. Add some more of the buffer. Wash it again. Mix it up. Spin it down. Remove the buffer. And that's basically what we're doing for the 2 washes. But these ones go pretty fast compared to the other one. Let's see. I'll show you. Much faster. Alright. Yeah, see, it's already getting there. It's a little easy, but real quickly it'll be clear. Like just like, I want to say like 20 more seconds or so. Be ready to clean up. And that's just because the previous buffer that we just took it out of had that polyethylene glycol in it, so it made it viscous, so it was a little harder for the beads to just travel through to the magnet. But this one doesn't have it, so it's a lot faster. Alright, so this one's ready to go. Alright, now we're just going to add another 150 microliters. This will be our second wash. So after this, we're going to add elution buffer. And what that's going to do is it's going to pull the DNA off of the beads. And we will get ready for actual sequencing on the flow cell. So we're all done with this. Close this up again. I'm just gonna close that. Mix it up, just like before. You don't have to do the whole way, just the majority of it. Spin it down. Spin it down. Briefly, just so we have everything basically collected towards the bottom. I know it's off camera, you can't see it, but we have a little mini centrifuge. Stick it back on the magnet, and this time I'm going to try to get as much of the buffer off as possible. So it's a little more intensive. And then while we're waiting, so after we add the elution buffer, we're going to do 2 10-minute incubations to try to get the DNA off. One's at 37 degrees Celsius, the other one's at room temperature. And that usually gets our library off of the magnets. But while we're doing that, we need to get tubes ready for quantifying our libraries, so we'll do that. And also get tubes ready for setting up the flow cells, because I need to mix a couple things into our flush buffer for the flow cells. And I need to get those tubes ready. Because once we're at the elution, we're basically just going to get the flow cells ready and load the samples onto them and stick them on the mid-ions for sequencing. Alright. And then what I'm going to do is I'm going to spin these down again just to make sure we get anything left as far as the buffer is down at the bottom. And then remove it with one of our smaller pipetters. So I want to get as much of this buffer off since it could interfere with the sequencing. All right, put that there. I'm gonna go spin these down in our little mini centrifuge. Then what I'll do is I'll stick them back on the magnets and pull off any of the residual buffer. Alright, so you can see there's the beads right there. I'm going to use our small P10, try to keep it close to beads. Pull off. Yeah, see, it's a small amount. Small amount of the buffer that was left at the bottom. Go. And this should get rid of any of that residual buffer, so we don't have to worry about that interfering with downstream sequencing. Yeah, there's usually like a microliter or 2 left over, although this one looks like it has a little. Oh, I had more. It's all good, that's why we do this. See, you're hanging out there. Right, there we go. It was right in the corner. I could see that little volume right there. Alright, let's change this to 31. Because we use 1 microliter for doing the quantification, so I can have an even 30. And now we're going to add the elution buffer, which is right here. I'm going to make sure that the beads are resuspended. And we'll start our first 10-minute incubation. And that's going to be at 37 degrees Celsius. Alright, let's close these up. I usually do this, and then I'm going to also spin them down. Make sure all the beads are within the elution buffer. Let's go spin these down, and I will do a little more resuspension. It's just a couple of more shakes. There we go. Oops. Sometimes I get stuck in here. There we go. And I'm just looking for the majority of the bead fillet to be resuspended. But I'm not doing it too vigorous because, like I said, we don't want to fragment our DNA. Trying to keep them long. Alright, that's good. Okay, I'm going to go put these at 37. Put some stuff back, and then what I'm going to do is I'm going to get our quantification tubes and buffer set, as well as the flow, the flush buffer for the flow cell set. So hang out with me on that. Alright, so the first incubation is done. Let me get my other glove on. I'm going to go get our samples, and then it'll be another 10 minutes at room temperature, and we'll be able to pull, hopefully, the DNA off of the beads. So let me go do that. Okay, alright, we are back. This was our last incubation for getting the getting the DNA off beads. We need to sing up for the next set here. Alright, so now what I'm going to do is we're going to get these back on the magnet. I'm going to move these over and make sure I have them in the right order. Yes, I do. So, this time, so the beads are going to the magnet again, but we're going to actually keep the liquid because that should be our DNA. Hold that right there, and then we're going to stick them in a new tube because we don't need the beads anymore. Let's move this down so it's easier to get to it. And like, go real fast, goes pretty fast. Then I'm going to quantify it, see how much DNA we have in these libraries using the same machine that I used earlier, so that's why we set up tubes beforehand. Look at that, perfect. Ready to go. Ready to go. And then I'm just gonna hold it closer to the magnet so I don't get the beads. Okay. And we just throw that away. We're done with that. We don't need them anymore. Because we should have our DNA right here ready to go. There we go. Alright, throw that away. I'm using our homemade whiteboard tips to do this too, so we don't fragment the DNA at this stage either. Not so late in the game. No fragmenting this late in the game. Alright. Put that away. Let's grab these two, although this one's sticking off. Let's grab this one. This one's ready. This one doesn't want to go. I'll have to let that one sit for a little bit longer. Alright, let's see. Sometimes if I just move it around. There we go. There we go. Much better. Much better. Okay, now I can grab it. Perfect. Perfect. Alright, that's all of our DNA off of the magnet. So, what I'm going to do next is take a microliter of each of these and stick them in tubes. I'm going to move these two down here. Spread these out so they're where they should be. And move this down a little bit. There we go. Vortex these once we have all DNA in the tubes. Let them sit for 2 minutes to let the dye for the quantification incorporate into the DNA. And then I will measure it on our machine. Alright, while we're waiting for this, I'm going to put in our sequencing buffer. Because this will be what we're going to basically put our libraries in for sequencing. Each of these tubes need sequencing buffer. There we go. And then these will be all ready to go, and I can put this away. Alright, so now that that's done, I'm going to go over to the machine, check all these out, and then I will be back. Alright, I'm back. So I measured all of our DNA samples, so now I have to do some math. to figure out how much we're actually going to be loading onto the flow cell. So let me figure that out. Alright, crunch the numbers. Now it's time to get everything ready for the actual library. So first thing is we need to add any water. Call it water, but it's actually this library solution which acts as water to bring it up to the correct volume for loading. So we will start, let's start at the top, which is 8.6 for 1. And that'll be for... this one. And then we have 7. And then 2.1 for the last one. Alright, so that's done. Done with that one. Alright, now I'm going to add the actual library. Top one, 99. This one. Alright, it's done, and then next would be 3.4. Alright, these two are done. Now I have to mix equally the one that we had 3 different regions set. And then the other 2 are 2 each. Love it when it's nice and even. Alright, so the libraries are all set now. We're all done. Move this off to the side just because it's easier. Ooh, I need it. What's up? We already got the DNA out. Okay, there we go. Alright, so this can move over here as well. So now what we're going to do, put that off to the side, is we're going to get the flow cells ready to go. There. So these are what the flow cells look like. I'm just pulling them out. This is what the flow cells look like. We're going to be loading 3 flow cells today. We're doing 3 different sequencing instructions. Alright, so first things first is we open up the little lever to get to the waste port. And I've set my P1000 to 200 because each of these has a bubble that we have to get out. And we want to get rid of the bubble because if there's any bubbles that make it into the actual sequencing port, it will destroy the pores, and we don't want that. So sequencing happens where this yellow is. So I got that one. Let's get this one. Almost got them there. Alright, that one's done. Let's get this one. There we go. Alright, now that one's set. Okay, so all these are set. I'm going to put this at 800 microliters. And so for each of these, we're going to, remember the flush buffers tubes that we made, going to put 800 microliters of flush buffer into the waste port. Make sure there's no bubbles on your tip, so you don't introduce bubbles. Do it slowly. There we go. That one's done. Let's do the next one. No bubbles. Alright, that one's done. Now for the last one. And then these are going to incubate at room temperature for 5 minutes. Again, no bubbles. Maybe easier. Now you can see when I do it this way. Alright, let's incubate this for 5 minutes. This step maybe better. Maybe not. You see now? So, not really. It's because I'm in the way. Sorry, I'm in the way. Alright, so I'm going to bring this down to 200 because we're going to need to set 200 for the step after the incubation. While we're waiting, let's add feeds to our libraries that I just loaded. So here's what the feeds look like. You might be able to see it. They're white in color. What we're going to do is we're going to add 25 microliters of feeds to our libraries. Perfect. And that's done. Don't need this anymore, don't need this anymore. I'm gonna close these up and then mix it. So you can see there's the beads right there that we just loaded onto there. So those are going to be ready to go. I'm going to set this at 75 microliters because that's how much we load the sample onto the flow cell. While we're waiting for the next 3.5 minutes, I'm going to go put our main libraries in the freezer, as well as get rid of those buffers that we don't need anymore, and then we'll be back. Alright, we're almost done with the 5-minute incubation. Which actually, let me line these up so I make sure I have them set to the way I know which library I put on which flow cell. There we go. Alright, all done. I don't need that anymore. Okay, so I'm going to open up the sample port by carefully pulling up on this little spot right here. And we want to be careful because we don't want to break off this little black nub right there into the sample port, and then we're not going to be able to load anything. Alright, so I'm going to load 200 more microliters of our flush buffer into the flow cell. Oops. Okay. Once again, no bubbles. And then I'm going to do this very carefully because if you might be able to see, but it's going to bubble up. And I don't want to lose too much of that liquid from bubbling up. So load it very slowly and carefully. And then we don't have to do the entire 200. We can only go to the first step on the pipette. It's right there. Alright, that one's done. Don't need that anymore. Let's do it for the next one. Okay, once again, no bubbles. Careful loading it as it bubbles up. And then, see, I went too fast on that one. It's fine, it'll go back down, but I'm trying to just not have too much go out. Nope. Don't need that one. Alright, let's do the last one. Okay. Once again, no bubbles. Be careful for the bubble. Okay. There. Alright, so that is all set. Now we're going to load the samples. Get this stuff out of the way, we don't need it anymore. Alright. I'm going to mix this again. I'm going to load all 75 microliters of our sample onto the flow cell. We do this in a dropwise fashion, right on that sample port. And through capillary action, it will get pulled down into where the membrane and where the pores are for sequencing. So there we go. I would say you probably can't see it on the camera. The camera's not detailed enough down here, but I can see as the beads go down into the actual membrane area. Almost done. There we go. Alright, that one's done. Load our second one. Same thing. Alright, now here's our last one. I'm actually going to mix it briefly just because those beads tend to... Oh, I'm stuck at the top. There we go. I love that when that happens. Alright, there we go. Okay. Last one. Oh, this one loads very nicely. Oh, look at that. Goes down a lot quicker than the other ones. Or it was. No, not so much. It was loading nicely. still faster than the other two. There we go. Alright. These samples are all loaded. Put stuff back. And they don't need anymore. I'm going to write on here so I know which samples they are. That one. And then this one. Alright. And what I'm going to do is we're going to cover these all up again. Once again, being careful, don't there. Cover up all the ports. There we go. Flip that away. That one. There we go. Okay, so these are all set. They're ready to be placed on our sequencer. Fortunately, I can't show you putting it on the sequencer. It's over at our other computer. But after I have these all set on the sequencer, what we're going to do is we're going to raid our actual sequencing channel, which is SeqLab on Twitch, and that is going to show you real time what our sequencing is, how our sequencing is going. So, but I will be back once I finish putting these on the sequencers, and we'll end stream and raid SeqLab. Alright, well everything is all set on the sequencer. Things are going, so I'm going to raid the channel. We'll end stream here. I want to thank you all for joining me today. If you want to see the VOD or my past VODs at all, I have a YouTube channel. Feel free to join that and look at it. Oh, I should probably change that. It's a much better link now. But yeah, so I appreciate you here. I hope you like the chill vibes as we got some targeted enrichments going today. But hope you join me next time and enjoy the rest of your day. Let's go raid C-Lab.